In this lesson, we'll review how neurons operate using internet animations. So we'll start with the synapse. We have a terminal here. We have synaptic vesicles, and in the vesicles are neurotransmitters. These are the chemicals that need to be released from the terminal. So when an electrical signal arrives at the terminal, the vesicles fuse, neurotransmitters are released, they diffuse across the gap, the space here, they bind to protein receptors on the membrane of the target cell, open channels, ions go rushing in, and that's an electrical signal in the target cell. If positive ions go in, it's going to tend to excite the cell. If negative ions go in, it will tend to inhibit the target cell. Over here on the right, then we see an animation. You'll notice up here we see the electrical signal arriving, vesicles fusing, transmitters released, and channels opening on the target cell. It's very quick. So we'll pause it at different places and break it down. Okay, as the electrical signal, the action potential, arrives at the terminal, another set of protein channels on the terminal membrane open up and allow yet another charged particle, calcium ions, into the terminal. The inflow of calcium triggers a series of events that moves the vesicles towards the membrane of the terminal. And so here we've caught one of the vesicles in the act of fusing, and it will be dumping its neurotransmitter, and you'll see that as red stuff coming out. Now, the red neurotransmitters have diffused across the synaptic cleft, they call it, and these yellow channels were closed a moment ago, but the neurotransmitter links up like a key in a lock and opens up these channels, and through these channels, ions will flow into the target cell. So this would be the spine on a dendrite. And so the yellow coming in here would have been the electrical current, the ions rushing into the target cell. Action potential arrives, vesicles fuse, transmitter released, channels open, ions into the target cell. Okay, we're ready in this animation to look at what happens in the axon. How does the axon carry the electrical signal down its length? First, we'll zoom into the brain and then see neurons. So here are individual neurons, and when they flash, like that one just flashed, that would be the generation of an action potential that races down the axon. Notice what we have here is we have a length of the axon, and they're putting these little particles outside the axon. Those would be sodium ions in the solution outside of the axon. And when the action potential races by, what's happening is these sodium ions are being allowed into the cell through those channels that stud the membrane. They're not showing us the channels, and they're not showing us potassium ions that are inside the axon. So they're focusing on sodium. But what the electrical signal is, what the action potential is this local inflow of sodium ions, but it's done in a kind of a sequential fashion. So first you'll see sodium ions enter here, and then these ones will enter, and then these ions will enter, and then these ions will enter. That's the electrical disturbance. Now, they can't get through the membrane initially because the sodium ions are closed. But when those first sodium ions open up, that's going to trigger the sequential opening up of all the rest of the channels down the length of the axon. There go the sodium ions into the axon. Then they have to be pumped back out so the neuron can make another electrical signal. And here we see the cells communicating with each other, and now we arrive at the terminal. So now we see the axon terminal, and we have several things going on here. So we've got the neurotransmitters in the vesicles. Sometimes you'll see the vesicle fuse with the terminal membrane and release the little red chemicals. Those are neurotransmitters. And then those neurotransmitters will bind to the channels, the protein channels on the membrane of the target cell. When those neurotransmitters bind, it causes a shape change. The channel opens up, and then the blue objects here would be charged particles, ions like sodium, rush in through the channel, and that's an electrical signal in the target cell. Here's another animation of the sodium channels and the potassium channels. Now when the sodium channels open up, you see a little inflow of sodium, indicated by this little color. And then potassium channels, when they open up, you see the potassium, the purple kind of leaving. It's the inflow of sodium that's going to trigger the nearby potassium channels and the next set of sodium channels down the length. Now these two objects here are little pumps. They're going to be restoring the ions to their previous concentrations. So when sodium rushes in, it has to be pumped back out. And likewise with potassium. When potassium leaves the cell, it has to be pumped back in so that the neuron can generate another action potential. Sometimes you'll see the action potential represented by this type of curve that you might record using electrodes. And so when we play the movie here, we'll see that when sodium enters, we get this part of the upper part of the curve, and when potassium leaves, we get the downward part of the curve. So when positively charged sodium enters the cell, that's this part of the action potential signal as recorded by electrodes, and when potassium leaves, that's this part of the action potential signal. And this animation nicely shows sort of the idea of the domino effect, the sequential opening of the sodium and potassium channels, which moves the electrical disturbance down the axon. This is the language of the brain, these electrical signals called action potentials. And so in this animation, just to wrap it up then, we've got all these dendrites out here that are receiving synapses from other neurons, and the little red little flashes here would be electrical currents that are generated at the synapses that are spread around the dendrites. Well, if they add up to reach threshold and pass threshold, then an action potential will be generated. So remember, out along the dendrites here at the synapses, chemicals are being released from axon terminals, channels being opened, ions go rushing in. That's the electrical signal. And what the cell body's doing, in a sense, is adding up all of those little electrical signals to see if it passes threshold. If it passes threshold, these first sodium channels will open up on the axon, and then you'll get the electrical signal racing down the axon, and we call that the action potential.